John Keith, thank you very much for having me here. Indeed, I'd like to introduce to you a new topic today. Let me give you the basic message at the very beginning. We're starting to see, and this is at its very beginning, a new development in medical research. Yet it's very old. And the basic message I'm going to give you today is that we poor human beings, as well as many other creatures on this planet, suffer from one thing. We have to digest. And the very same enzymes that we use and the very same system that we use to digest food serves to digest ourselves. So we auto-digest. That's the message I have for you today. So now, I know it's not very convincing. You may say, oops, maybe there is something to that. But it's all important that you don't believe me one word that I'm going to say, and just listen to the evidence and try to poke some holes into that evidence. I have a conflict of interest because we're trying to translate these ideas into a therapy. I'll explain to you as we go along. And of course, I have a conflict of interest because you can't do this research without money, because you have to support all of these people that you see on there. And there's a local team in San Diego, and there are collaborators across the country that have participated in this work. You probably may recognize one or two names on there, but I'll explain more in detail as we go through these slides what the story is and who has participated in what fashion. So, the starting point, the departure for this discussion this morning is a phenomenon that I have had the opportunity to observe for the last about 2 or 3 decades. And that is, this word inflammation has appeared in the literature, and it's now in the public literature. And physicians and medical students learn about this. The word was originally introduced by a pathologist about 1822, and it says that there is a burning feeling when you scratch your skin. Well, now it's been shown, in especially the last 20 years, that every human disease that's ever been looked at in clinical trials, is no longer speculation, is an inflammatory disease. And in very broad terms, I know it's a little oversimplified, you can divide this into 2 basic directions. One is infectious diseases. Indeed, if you come in contact with such a person, you have a good chance that you're going to catch them, and there are various reasons for that. But then there is a very large body of non-infectious, non-communicable inflammation. You can be sitting close to somebody with atherosclerosis all your life long. You're not going to get atherosclerosis from that person. You can sit close to somebody who is aging, or something. You're not going to age more than that. And I'm going to talk specifically today about a condition, the worst condition, namely sepsis and shock. You can be in an intensive care unit. You're not going to go into a shock situation. So there are ways in which we can generate in our body an inflammation which is not due to these mechanisms. If you ask yourself, what is this phenomenon? What is nature telling us? Well, one thing you have to recognize is that physicians measure the level of inflammation by taking a blood sample, and they measure things like C-reactive protein or the interleukin-6 or many other chemical parameters. These molecules are all generated mostly in your liver, some in the endothelial cells, and others, in order to repair the tissue. These are repair molecules. So fundamentally, the inflammatory cascade, as we like to call it, because it's a long sequence of events that nature goes through in order to repair a tissue, it is really a wound healing process. All these molecules that we express and that are pro-inflammatory are in fact repair molecules. And so if you can accept that notion, then in that particular case, you're justified to ask the question, what causes tissue injury? Okay, we talked about the possibility of infections. There's a very broad range of possibilities for inflammation and tissue repair due to trauma, all kinds of traumas, obviously. Our body is very intolerant to temperature changes, up or down. We just have to live around 37 degrees. If we go much above or below that, we run into a condition that causes tissue injury and will cause an inflammatory reaction. And same holds true for all kinds of chemical reactions. It's a long, big category of reaction. Now, if you talk to clinicians, this is the list they will eventually point you to if they are talking about a patient who has an inflammatory marker, like C-reactive protein. And I have been thinking about this question for quite some time and start to realize that something isn't quite right. And one of the ways this came about is by looking specifically at the shock situations. If you go into an intensive care unit and you look at patients at shock, they are severely ill. They may be passing out before the end of the day. And yet, it's often very, very difficult to show that there is a participation by any of these mechanisms. We can do experimental animal studies, for example, hemorrhagic shock. There's no compelling evidence, in spite of a large number of trials, that anything like an infection is involved in the death of these animals. So this led us then to a line of thinking in which we said, well, in these individuals, one of the things you find is a form of cell activation that we test in the laboratory by looking at the transformation of these are circulating leukocytes, into these activated states. And the remarkable thing about this process is that this happens in the circulation. And these cells, as they circulate and go through capillaries, they have great difficulties. And so from a microvascular point of view, such a transformation is a very serious transition. It goes hand in hand with what the clinicians see, like an elevated C-reactive protein or IL-6 expression or TNF-alpha expression, all kinds of markers that you can get. It's a marker for inflammation. And so about the year 1999, we asked the question, if in an animal that you subject, for example, to hemorrhagic shock, there is a transition of a large number of cells in the circulation from this state to that state, what could be the origin of that? And we did an elemental experiment that fundamentally transformed my thinking about human disease. And this is shown in this slide. So we took the extract of different organs, all kinds of organs you like to pick, and we homogenized them. It's a terrible thing for an engineer to do, but we did it. And we then asked the question, is that homogenate when we put it in contact with some naive cells, so with healthy cells, put these homogenates in there, would any of them actually activate one of these, would any of the homogenate activate one of these cells? And the experiment showed us is that we have to pay attention to the pancreas. It was a total surprise. That result we have now repeated, I don't know, almost 100 times. It's one of the most robust results in inflammation. We then asked the question, if you take one of the enzymes that's produced in the intestine, trypsin, which is a digestive enzymes, and you just mix it up together with any of these homogenates, you start to see that in fact every organ now, it's the red bars, is a producer of inflammatory mediators. Now you will say, hey, come on, digestive enzymes in the brain, that's a bunch of nonsense, okay? Forget about it. So for the moment, what I will do is I will say, let's look at the two important organs, that's the pancreas and the intestine. And you notice here on the intestine, what we actually had done is we had, when we took, this is fresh tested from a young teenage animal, when we took that intestine out, we actually rinsed it out. We washed everything out, and then we got the blue bar. And then we did the experiment again. We actually put the digestive enzymes back in there, or in this case, only trypsin in there. And suddenly we have a mediator that is as powerful as the pancreas. If you add digestive enzymes to the pancreas, it doesn't make a difference. So there is something about these digestive enzymes. I'll say something more. Now, we're forced to think about the intestines. And you ask yourself, that cannot be. Well, if you go back into the medical history, that is hundreds, thousands of years old. The intestine is at the source of human disease. Not only do these homogenates I showed you activate cells, but actually they are cytotoxic. Here's a set of experiments. These are our control cells. We expose them to a homogenate from the intestine, the same stuff that you just saw. And within about 30 minutes, you see there's complete destruction of these cells. We identified the mechanism for that. That turns out to be free fatty acids that are produced by lipases in a tissue homogenate. And the free fatty acids, remember, those are soap. It's the same material as soap. If they appear, it dissolves all lipid membranes, and therefore you see a complete destruction. I don't even know how to call this, necrosis, apoptosis, whatever name you want to use for that. So the focus is on digestive enzymes. They're produced in your pancreas. The pancreas produces insulin. It's about 5% of the engine, and 95% is cranking out digestive enzymes all day long. They can degrade every protein, every carbohydrate, every lipid, every carbohydrate other than starch. We cannot break down starch. And every nuclease, every nucleic acid sequence, irrespective of its source. When you eat a protein from a plant, or from a fish, or from a other human being, you have the ability to degrade that protease. You have, in addition to that, something very important. This is an experiment where, in a hemorrhagic shock protocol, we took a plasma sample at the worst possible time period, when the plasma had just reached maximum level of destruction. And we took, for comparison, a small amount of fluid directly out of the intestine. And then we made a measurement of the protease activity. This is expressed in terms of fluorescent units. And the way you do that is you have a little peptide that's fluorescently quenched. When it gets cleaved by the peptide of your interest, it becomes fluorescent. So you measure fluorescent units on there. And all I want you to pay attention to is the order of magnitude. The highest level we ever got of an animal is somewhere of the order of 20 units. In the intestine, we never start below 2,500. So if you ask yourself, if we had the ability to take a picture of a human being, where you see the enzymatic activity, you would see a red glowing intestine. And everything hopefully is blue or black. The question is really, what is it? So our intestine, our digestive enzymes are something to cope with. These are the most powerful enzymes in our body. And that raises, of course, the following question. Okay, as you digest, this food comes down your stomach. Right there is the pancreas. It mixes in the duodenum with the food stuff that's come through. Its pH is balanced. Everything is pretty much neutral. And then we go through this long ride to the small intestine. It's about 4 meters, 6 meters. And you have fully activated enzymes in there. So if you travel to Poland or Germany or Italy and you eat that intestine, you ask yourself the question, what has nature done that I don't digest myself? Or is this a red herring? How come I don't digest myself? That question has suddenly come into focus in my thinking. And what I'll tell you today is that's nature's ways of solving things. It's okay for a while, but then it fails. Okay. What's the mechanism that protects us from our digestive enzymes? Well, you have to look at the small intestine. That's one in cross-section. It's from a cat. And the place where you digest is right here. That's where your proteases are, where your digestive enzymes are, these very high concentrations. And the protection that you have from digesting that tissue, the mucosal layer, there's a smooth muscle layer that provides the peristalsis, is really derived from the layer on top of one of these villi. It's magnified here. This is an epithelium that sits all around. There are some specialized cells in between. They're called goblet cells. And this picture is an EM picture from Johannes Rodin. You have to look at it in a slightly different way with a histochemical label. Then you see that on top of these villi, is actually another mushy layer, a slimy layer on top of that. It's a mucin layer. And that mucin layer actually in the rat consists of two parts. One is the mucin that is coming out from these goblet cells. They actually secrete that mucin, and they allow it to mix with the food that comes down. And when you go potty, the slimy part is that mucin. There's a second mucin present, and that sits tightly attached to the epithelial cells. That mucin is all important because it protects your own epithelial cells from your own digestive enzymes. Because this epithelial cell is supposed to absorb, through various mechanisms and molecular transporters, the food that has just been degraded here and is supposed to be absorbed into your body to get the nutritional support. So we have multiple layers of protection in there, and the question comes out, just how good is this? Anybody among you who is studying the blood-brain barrier, who studies any other barrier, will know their barriers are never perfect. And there are all kinds of ways this barrier can break down. And I want to show you an example here. This is one of those zymographic images. Again, the orientation is here, the villi pointing upwards. Can we turn the light just a little bit down? Is that possible? Thank you. Thank you, John.
A lot down.
Yeah, that would be helpful, yes. This is the muscle layer here at the bottom. And this is a frozen section, so it's a thin section. We put on that section a substrate that will become fluorescent. It's the light blue once it's clipped. So the bright blue color is a measure for the enzymatic activity. And all we've done in this particular case, this is in a rat, we subject it to a splanchnic artery occlusion. So in other words, we interrupt the blood flow for a short period of time, in 15 and 30 minutes, and you see that this enzymatic activity enters into the wall of the intestine. As a matter of fact, it goes all across. Is this inconsequential? No. This is actually what happens. As these digestive enzymes come in, they digest away, starting at the tip of the villi, the tissue. The tissue becomes food, okay? And this degrades, and if you come back, say, an hour later, the entire villus structure is gone. That means the protection layer you have against digestive enzymes is now completely gone. And you're exposed now to your own digestive enzymes. These digestive enzymes don't stay just in the intestine. They actually make it into the plasma. So if you look at the levels, this is a hemorrhagic shock protocol at the beginning, and 2 hours later, you see there's a systematic enhancement. Not only do we find this for all of the three major proteases, this is elastase, chymotrypsin, and trypsin in the plasma, but we also find the digestive enzymes in the peritoneum. And in fact, I have additional evidence to show you that these digestive enzymes also escape into the lymphatics. So the way you can think about this is that every pathway out of the intestine is now being utilized under these hemorrhagic shock conditions for the digestive enzymes to enter the rest of your body. There's no pathway that is blocked. And you now have, and this is activity, these are actual enzymatic hydrolytic activities, you have actually a bunch of enzymes in your bloodstream that are capable of taking you apart. Everything from now on is really nasty. It's terrible. It's bad news. One of the things that these digestive enzymes do is they activate others. Here's an example. We were focused on matrix metalloproteinases, MMP9, and what we started to see is that both the protein level as well as the activity level, this light color that you see there, is again a measure for an actual activity in this plasma, is enhanced in these short periods of time. We find the same thing in peritoneal fluid. There's an MMP activation. And then if we look around among different organs, at that time we were interested in the lung, the liver, and the heart, we find that every one of these organs is suddenly showing not only the digestive enzyme activity, but on top of that, the MMP9 activity. And I can add to you, I don't have the data with me, that we also looked at the brain. The brain is also infiltrated now by digestive enzymes. Okay, there's a long list of papers that we published on how these enzymes actually make it into the bloodstream. And I want to move on today and talk a little bit for a minute about, is there anything we can ever do about such situation? And so the case we were interested in was shock. These are these patients who are in intensive care unit, and antibiotics don't work anymore. And physicians have utilized all of their abilities to interfere with the breakdown of organs that is taking place in these patients, called multi-organ failure, by putting them on a respirator, on a dialysis device, and so on. Physicians are at the end of the line. And about 20 to 30% of all patients who come into intensive care unit, they will eventually pass. So the issue is there something we can do about it if these mechanisms should be valid in humans? And the approach we took is said, well, why don't we expose the intestine? Here's the small intestine, just shown schematically. Here's the pancreas. And we introduce by local injections. We did this in a rat. The rat intestine is about 1 meter long. And so every about 5 centimeters, we made one of these local injections. It takes about 15 minutes. You're done with it. And then you ask yourself the question, can you block these digestive enzymes? We used a drug called ANGD that was developed by a Japanese company at the time in order to treat pancreatitis. It's not approved in the United States. It's approved in Asia. And it's specifically designed to block these digestive enzymes, trypsin, chymotrypsin, and elastase that I showed you previously. So what my technician, Frank, did is he took out an intestine. This is from the duodenum to the cecum, so this is this full length of the intestine. Cut it up into small pieces and then measured the proteolytic activity. And this is a control case where you can see that there is pretty much a uniform distribution of proteolytic activity. In other words, as you go along your intestine, if this translates to humans, there isn't a fall off or decrease of digestive enzyme activity. The full length of the intestine is fully active. If you now do this in a hemorrhagic shock case that I mentioned to you, all we've done is reduce the blood flow for about 2 hours and then reconstituted everything. It's all we have done to this animal. We have this large increase. And if we treat in this fashion by these local injection, this enteral injection, we're able to bring down the activity this way. We published a long paper on this story, and I would just want to give you highlights. If you block in the lumen of the intestine, the small intestine, what are you doing to this animal? The first thing you observe is there is a dramatic increase in survival rates. It goes from about 3 out of 12 animals we did at the time in the treated case to 10 out of 12 animals. And we were able to achieve that with different types of protease inhibitors. I mentioned ANGD. We used a drug that's approved in the United States, tranexamic acid. Protein is an older drug. It's no longer used. And in every one of these cases, you can see we can reduce the mortality. We can not only reduce it in the case of this hemorrhagic shock protocol, but we studied actually multiple other forms of shock, and we see a similar result. I want to show you a picture on how this looks like. So this is a normal intestine in a rat. This is the right heart, the lung. And this is what it looks like after these 2 hours of ischemia. You see this red coloring that appears on that. That's due to the escape of red cells through postcapillary venules, which are stuck in the tissue. You can't wash them out. And physicians refer to these as lesion formations. These cells are initially protective, but within a couple of hours, because of the presence of hemoglobin, they're actually cytotoxic. Now, if we, and this is typical for hemorrhagic shock or septic shock, they all look pretty much the same. If you now take an animal that has been pre-treated just by an injection of digestive enzymes into the lumen of the intestine, that's all we have done. We have not given anything IV or anything of that nature. We're able to reduce this injury in the right heart as well as in the lung, although not completely prevented, because this treatment was provided an hour after we had started with this hemorrhagic shock protocol. So there's a dramatic improvement of morphology, and there are many other studies we carried out to confirm that. And this is now in clinical trial. So there, as I speak, there are patients now being enrolled where we are trying the first time in medical history to see whether we can make a person survive by blocking that person's digestive enzymes as good as we can. And I hope that perhaps in a year or so I'd be able to report back to you. This would be actually the first treatment for such patients, because these have no treatment options today. Now, I told you earlier that there is a major destruction going on. And one of the most dramatic ways I can show you just how destructive this process is by showing you a scan. This is from about 15 kilodalton to about 25 kilodalton, a plasma scan of a rat plasma. There you see different types of peaks. This is mass spec. And each of these is a biomolecule that's well recognized, and they are part of the normal life of a young teenage rat. When we take that rat and expose it to this hemorrhagic shock that I mentioned to you, where you see this destruction in the tissue, this is how it looks like in the plasma. The only words I can find for this, there is a wholesale molecular destruction going on. And no surprise, this animal is actually dead within the next 2 or 3 hours. So these digestive enzymes cause a major destruction. Large number of fragments are being produced. It's the most dramatic destruction I've ever seen, actually, in any disease in this case. And if you look at individual, we did a peptidomic analysis. A normal rat has about 126 peptides. These are below 15 kilodalton. About 30 of which are unique to a control state. If you put the animal into this hemorrhagic shock, you now have 295 peptides. And what has happened is a large number of proteins have actually been degraded. So you go from 30 to 199. So it's another confirmation that the entry of digestive enzyme leads to a massive destruction of what's going on in your body. There are various categories of proteins that have been destroyed, including, for example, inhibitors for the digestive enzymes. They themselves are being destroyed. The destruction just goes on and on. Here's another example. One of the events that take place in an animal or in humans in the intensive care unit is that they develop what is called insulin resistance. That's exactly the same insulin resistance as in chronic adults, except in this particular case it's called acute because it happens within about 2 hours. And then physicians notice that the patient has an elevated glucose level, and they try to bring that glucose level down by administering insulin. They discover it doesn't work. In our animals, a glucose bolus, this is how we can test this, leads to a transient elevation, and then the glucose levels go back down to a low value. This takes about 2 hours. If you do the experiment in a slightly different way, where you give an insulin bolus, you see, of course, the opposite. You see a drop in the glucose levels, and then a slow return back to controls. This is how an animal in control state looks like. This is before shock. This is how it looks like after the shock. So this is the acute insulin resistance. There's a very large increase, and it overshoots, in fact, our experimental range. And the glucose levels never come down to controls. There is still a decrease in the glucose levels with an insulin bolus, but you see it's greatly attenuated. There's no longer this strong response that you would expect to see from a young, healthy animal. When we block the digestive enzymes in the lumen of the intestine, same trick again. It's a basic elementary transport trick. The blue curve it arises. We're able to essentially reverse this process. And this is due to the fact that if you label the surface epitope of the insulin receptor with an antibody against the binding domain where insulin binds, you discover that the density of these receptors, shown by the red fluorescence, is decreased after shock. It's not eliminated, but it's decreased. And it's maintained if the animal has been treated with a protease inhibitor beforehand. So the concept that was born out of these experiments is that a normal insulin receptor, when it binds to insulin and then signals to the inside for various linker molecules to the glucose transporter to incorporate into the membrane and allow glucose to get in. That's this well-known insulin transport mechanism. That mechanism, in the presence of digestive enzymes, is destroyed at the very beginning due to the presence of these proteases. That destroys this receptor. It cannot signal anymore, and as a result, of course, you develop insulin resistance. So what I've told you so far is from these experiments, it's just a short overview of a much larger set of studies, that the breakdown of the mucosal barrier in the small intestine, I showed you the example for the ischemia, leads to leakage of pancreatic digestive enzymes and inflammatory products they produce. I mentioned the free fatty acids and peptides they form. Leads to activation of secondary degrading enzymes like the MMPs. It leads to autodigestion. The intestine just becomes digested itself. You have receptor cleavage, and as a result, you have these acute cell failures. And you eventually, of course, go on into multi-organ failure and death. If you inhibit the proteolytic digestive enzymes, all of these are inhibited, all at the same time, telling you this is the origin of the problem. This is at the very beginning of what goes wrong in these animals. Everything else here are symptoms. If you want to interfere with the beginning of this process, you better block the digestive enzymes. Okay, so much for the acute shock situation. This is the most dramatic case can ever happen in your life. I don't wish it any of you. Not until we have a good treatment to block your digestive enzymes. Wait for that day, okay? In the meantime, however, you folks have a very good feeling for biological membranes. I started to ask the question very soon, oh, wait a second. This is a transport problem. This is a biomechanics problem. This is a biomechanics problem to one of the most important events in our life from here on forward. The question is it possible that digestive enzymes could ever leak right now? Okay? Is there a possibility that as I go through life and I do certain things, not knowing, I may actually have an escape of my digestive enzymes? And is there anything good about that? Should that be? Is there something I need for that to happen? Or can I be satisfied with my digestive enzymes just in my intestine and nowhere else? So to make it again very concrete, what we did is we took an animal model, which is called the spontaneously hypertensive rat, SHR. The NIH has invested a large amount of money into this animal, mostly to find out why does this animal have this elevated blood pressure. This is over a lifetime, over half a lifetime. This animal develops a high blood pressure. There is a low blood pressure control. It's called the Wistar-Kyoto rat. I call it just Wiki. So I'm going to talk about SHR and Wiki. This animal has a lower blood pressure. And both of them are actually derived from a Wistar strain originally in Japan by Professor Okamoto in Kyoto, who originally had this idea to develop this model. It's a genetic model, and it's been celebrated for its elevated blood pressure. But I had the opportunity to study this animal at the microvascular level for many years. And I come away with the idea that this is an illusion. Wait a second, folks. Elevated blood pressure, yes, it has that. But if you really study various aspects inside this animal, you find out this animal has what we call in humans metabolic disease. All kinds of other problems. There is an endothelial apoptosis, and as a result, the capillary network, if you look at small blood vessels, they disappear. So if you look into the eye or different parts of it, these patients have actually a reduced number of capillaries. You can look at their insulin resistance, and they have an insulin resistance. A mild form, but they have an insulin resistance, just like humans. In humans, you can't tell, is this person hypertensive or diabetic, or is he diabetic and hypertension? The two of them are mixed up with each other. You can look at a mechanotransduction, a very fascinating element. It is defective in this animal. Still present, but defective. There's a major immune suppression, and the animal can't sleep very well, just like humans. You wake up in the middle of the night, and you're bright awake, and you can't fall asleep again. This animal has exactly the same problem. So the issue is, from an engineering point of view, okay, focusing on elevated blood pressure through looking at arterial constriction is interesting, but you can't look at this animal as a biological entity without looking at these additional events. So I'm going to present to you now a concept that embraces all of this under one roof. Once we saw the results in shock, the first experiment that I did with my my long-term co-worker, Frank Delano, and said, Frank, could you please measure the protease activity in the SHR just in the plasma? Just take a bit of plasma and measure protease activity. And this is what he came up with. And I show you this boring graph here because I want you to recognize the numbers. This is the Wistar rat. That's where both of these strains come from. This is the normal, low pressure control. This is the SHR. And if you look at these numbers, I'm not talking about an order of magnitude increase as we had it in shock. This is only about 20, 30% at most. 30% is high. It's a 10, 15, 20% increase. So you really have to have quantitative techniques to get at those numbers, okay? When we looked around, what could these proteases possibly be? We find out that MAPs are very prominent. There are certain serine proteases. Again, I show you these curves here just to show you there is an elevation, but it isn't a qualitative experiment wouldn't tell you. You have to be really quantitative. There's also an inhibition of the inhibitors of these proteases. We identified, for example, this inhibitor TIMP1, and it's decreased in the SHR. I'd like you to take a picture, have a visual picture of how this actually looks like on a tissue. So this is a fresh tissue, it's a mesentery. Here's a venial, here's an arterial, a bunch of little capillaries in between. And this is the wiki rat. And this is a substrate that's clipped by MMP1 and MMP9. This is how the wiki looks like. This is how the SHR looks like. So it's comparable. But there are, first of all, these punctated locations inside certain cells where you find this activity. And overall, there's an increase in there. Now, since it was an MMP, that was great news because there are, in fact, companies that are producing MMP inhibitors. They were originally developed for the cancer research. They all failed in cancer research. And we went back and said, guys, could you give us a little bit of your MMP? So we treated our SHR with an MMP inhibitor. We used doxycycline because that's approved in humans. And doxycycline, by the way, is an antibiotic, but at low concentration it's an MMP inhibitor. So you see how complex drugs really are. And we treated these animals for 24 weeks. And this is what we obtained as in comparison. Again, you have to make quantitative measurements to come up with a reasonable result. And I want to show you again the numbers. See, we're talking about at most an increase of about 20, 30%. So if you want to measure this in humans, you have to have a real good a technique that resolves this type of difference. Now, I told you that when digestive enzymes activate MMPs, one or both are able of clipping receptors. Now, in the SHR, one of the most important receptors is the beta-2 adrenergic receptor. I remind you, that's the receptor that binds hormones that are coming from your adrenal gland. And they are responsible for dilating the artery. Okay? These are dilatory stimuli with epinephrine, norepinephrine. And we've known from pharmacological studies for a long time that these beta-adrenergic receptors don't work very well. So what Federico Rodriguez did is he took an antibody against the N-terminus, that is the external domain of this beta-2 adrenergic receptor, and he labeled it. He did this on the aorta of a Wistar, a wiki, and the SHR. And what you see here is the labeling. This is the same antibody. The labeling is greatly attenuated on the SHR, giving us the first evidence that actually what's happening in this animal, that its own digestive enzymes destroy that receptor. And as a result, of course, the ability of this animal to dilate is gone. He then took an antibody against the C-terminus, so that's on the inside. Repeat the same experiment and we find full labeling. So this is now direct evidence that actually what has happened in vivo, this is not an in vitro result, that these digestive enzymes, or the MMPs, we don't quite know which one, have actually destroyed that receptor. And that is the cause for the vasoconstriction. We had to do lots of control experiments. I won't have time to show them to you, but it was very clear that there was a enzymatic destruction of this receptor that affected the outside, and there was a very large turnover. Now remember, I told you, people with elevated blood pressure also have insulin resistance. The SHR is the same. So we asked the question, if we use an antibody against the insulin receptor that binds to the extracellular domain, how does it look like on the wiki SHR? Here you see those measurements. This is on a connective tissue. When we treat these animals, this is 24 weeks, in the same experiment I showed you previously, we see that in fact these insulin receptors are restored on the SHR. Telling us now that the same enzymatic destruction that leads to the loss of the beta-2 receptor also leads to the loss of the insulin receptor. And the interesting question then is, does the same treatment against MMPs help with doxycycline? Does it help to cure both conditions? And the answer is yes. Here are the numbers. The typical blood pressure of the SHR is about 175 millimeter mercury. And after this treatment, they drop to 126, which is about the value of its low pressure control. The low pressure control, by the way, also drops off, interesting enough. Now let's look at the glucose level. Same animal, same experiments. There is a mild form of hyperglycemia. It's also noticeable by a glycated hemoglobin measurement, which is controlled and which is reduced to control values in the SHR after this treatment. So one single treatment to block the enzymatic activity has helped both conditions through a single mechanism, namely a prevention of this destruction of these receptors. Now comes the question. I told you there are lots of other problems that this animal has. One of them is the loss of capillaries. This shows you a little piece of a mesentery. This is an actual reconstruction. And this is in the SHR and this is in the wiki. There's two different length scales. The SHR just has, as I mentioned already, this very flimsy capillary network, barely any capillaries. It's a dramatic reduction, was already discovered in the 70s. When we treat the SHR with doxycycline, we see a regrowth of the capillaries, not quite to the level of the wiki, interesting enough, but there is clearly a reconstruction. We looked at the cause for that, and that was due to the fact that in this enzymatic destruction, a third receptor that's being destroyed is the insulin growth factor receptor, VEGFR2. That growth receptor is responsible for the lack of apoptosis in that cell. If you block that receptor, which was originally designed for cancer treatment, here it happens through an enzymatic process. In that particular case, VEGFR2 can't function anymore. So the concept is now becoming clear. What is happening here is that we have this uncontrolled protease activity, all these little Chapmans. They are destroying the outside of these receptors. And therefore, the normal signaling mechanism like arterial vasodilation or glucose transport or the endotheliovascular growth factor signaling is all interrupted. Instead, we end up with arterial constriction. We have a defective glucose transport. We have endothelial apoptosis, and all of these physiological functions are attenuated. So we have now the first time a concept that explains to us, or helps to explain, why we have so many comorbidities. And in fact, we went on to study now a wide range of receptors. This was literally one receptor at the time. We showed it was clipped. We showed that after the clipping, the receptor could be restored by blockade of the enzymes, and we showed which enzymes was responsible for that. So this is a long list of papers that we put together. I wrote a summary report about it if you don't want to read them all individually. But it's conceptually always the same thing. So besides these three receptors I just mentioned, we showed that the FMLP receptor is being clipped on the SHR, and that's why we have a suppression of the shear response. The FMLP receptor is a shear stress sensor on that cell, and it's being destroyed through the same enzymatic mechanism, and therefore this cell has a very poor shear stress response. It also has a very poor chemotactic response to certain peptides because the same receptor is being used in nature to activate the chemotactic response and the pseudopod projection that goes along with it. But this is not limited at all. The leukocyte adhesion receptors, beta-2 integrin, ICAM1, P-selectin, PSGL1, they're all clipped. And as a result, this animal has a defect in leukocyte adhesion. As a result, you have an immune suppression. The scavenger receptor CD36 is reduced in its expression on macrophages. The red cell glycocalyx are clipped on the SHR. And I'll show you in a minute, even this cerebral serotonin receptor is reduced, which is associated with a reduced sleep insomnia. All of these physiological deficiencies are repaired if you block the digestive enzymes. You don't need to go to different clinics. You take one protease inhibitor, and that will restore, at least for the SHR, this entire metabolic syndrome. So here, I know you guys are interested in the brain. I wanted to show you an example of this. This is a antibody, monoclonal antibody against serotonin, the T1 receptor. This is the brain of a wiki, SHR. You see this reduction of intensity, and this is after the 2-week treatment, 20-week treatment, a restoration of these receptor densities. And these are the measurements in there. That goes hand with a loss of sleep quality. The SHR is characterized, we did this experiment with Terry Koo in Taiwan, who has a telemetry to monitor these animals. And what he shows is that the transition from deep sleep into quiet sleep, deep sleep to quiet sleep, which is very slow in a normal, healthy animal. They just sleep very deep. They don't worry whether the door opens or not, is no longer present in an SHR. In SHR, you just look at them, and they immediately go back into a shallow sleep, into this quiet sleep. So then the frequency of transition from deep sleep to quiet sleep is greatly enhanced. If he treats the animals with doxycycline, this is again restored, consistent with these experimental results. So you have direct evidence that these digestive enzymes and their mediators are present in the brain. And here's a second experiment that's ongoing at the moment. This is the leptin receptor, OBR. It's immunolabeled in the hypothalamus, close to the third ventricle. This is the immune density that you identify for that receptor in this particular domain. And this is what it looks like in an animal that has been fed for 12 weeks with high-fat diet. So this is a different model. And here's the direct evidence that, in fact, high-fat diet is potentially associated with an activation of degrading enzymes in the brain. This, if you treat the animal, if you knock out one of the MMPs that we identified is critical in this experiment, MMP2, by using an MMP2 knockout, the weight gain that these animals undergo in a 16-week period is significantly reduced. So what I told you is that the SHR and rats with high-fat diet have an unchecked proteolytic enzymatic activities in the circulation. This proteolytic activity of membrane receptors in the metabolic disease is a mechanism for multiple cellular clinical comorbidities. And this whole idea, as I mentioned at the beginning, really needs to be tested at this particular point in humans. Chronic inhibition of proteolytic protease activity serves to attenuate membrane receptor cleavage and maintain cell functions that they carry out. So every receptor that you lose through this mechanism means a loss of that function, and as a result, you will develop a morbidity. So where are we? Well, what I've tried to do today is convince you that it's worthwhile to think about. Don't believe anything I said. That it's worthwhile to think about that the assumption that your digestive enzymes are always in your intestine may be wrong. Okay? And among these different injury mechanisms that we discussed, really one has to add another one, and that's autodigestion. And if autodigestion is present in all these non-infectious inflammatory conditions, it may turn out that the autodigestion is actually more important than the infectious disease. This is something that will profoundly influence our existence here in life. It all remains to be seen. Finally, I'd like to conclude and say, is there any evidence in humans for that? Well, as I mentioned, it's very flimsy at the moment. It's the time to get involved in this field. What we did is the following. I mentioned to you we have to have much more precise methods to measure protease activity. And what I did is I worked together with Mike Heller, who is the founder of Nanogen and an expert in high-precision diagnostics. And he proposed the following approach. You take a very small drop of blood, finger prick, and you place it into a little well that sits over a gel. And in that well, you put whole blood in there, so no separation necessary, no processing at all. So it's clinically useful, okay? And then we add into this blood sample a peptide that is just like all the other peptides I showed you. It has a fluorescent end and has a buffer on the other side, but the sequence of these peptide is selected that after it's being clipped by the enzyme, in this particular case MMP2, it falls apart into two parts. And the two parts change their charge. So the peptide is specifically designed so that the fluorescent part of that peptide is now positively charged. The rest is negative charged. So once we allow this reaction to occur for a couple of minutes in a gel like this, we turn on an electric field, and that pulls, with a negative electrode at the bottom, that pulls all these positively charged fluorescent peptides into this gel. So I can see at the bottom in the gel now the reaction product that was capable, that the blood was able to produce. And this is a method that measures the protease activity about 2 to 3 orders of magnitude more precise than all other methods currently available. And you can do it on whole blood. You don't have to separate anything, and the whole thing can be done in just a few minutes. So it's what we call point of care type of measurements. We used this measurement, and the graduate students were very interested, Augusto and Elaine, and asked the question, okay, if there's any truth to that, so this is what I just told you, we should be able to demonstrate it in young graduate students. All right, okay. I said, okay, we're going to go to McDonald's, I pay the bill, and we're going to have a high-fat diet. So it's Turbo Mac, some chocolate, orange juice, and I think some potato chips. It takes about 15 minutes to eat. And then we take a blood sample right at the beginning, and then about every half hour, and this whole thing goes out to 5 hours. This is what they came up with. This is the young, healthy graduate students. This is a significant increase in proteolytic activity. It's very transient, and then it sort of flux on. If you don't eat breakfast, this is what it looks like. This is a small group. There's only about 10 or 12 individuals in group, so it's just a pilot study. This is what it looks like if you have somebody who's pre-hypertensive. Pre-hypertensive means you've just been diagnosed with an early form of hypertension. You're not taking any hypertension, diabetic medicine. I'm sorry. So pre-diabetics are up here. They're elevated. True diabetics are elevated. They're on medication. So is high up. It's even elevated again, transient during this meal, but then they never really quite fall down to the control values. And there's a very complicated curve which we don't understand at this point. But the point I want to make is if you want to measure proteolytic activity, there are two things. Number one, you need a very sensitive technique. Number two, you need to be able to catch these transients. In other words, it is the exact opposite of, for example, C-reactive protein that many of you probably know your C-reactive measurement from your last visit with during a checkup. That value is very stable during the day. It's a repair molecule. These are just the opposite. They fluctuate all over the place. And so as a consequence, we really have to introduce a whole new set of technologies to assess this. This is a last day's value. We did many other protease measurements, and there are similar fluctuations in there. That protease activity is really consequential. If you look at the same time at the insulin receptor. And this is an experiment where we took that same plasma you just saw and put it on naive cells. And then we label with an antibody the insulin receptor. This is how full labeling looks like. And this is how it looks like over the time course. These are representative examples. Here's the measurements. You see there is a reduction of the insulin receptor. In other words, this plasma clips the insulin receptor, your insulin receptor. This is true for the controls. In the control fasting, it doesn't occur. In the pre-diabetic, there is already a very low value of insulin receptors. And in the diabetics, it's actually interesting, it's slightly elevated. We don't fully understand how these curves, why they come out the way they are. These are just some initial pilot studies. So, I thank you for your interest. I hope I could inspire you that there is another fundamental mechanism that we completely overlooked that may lead to destruction of our body. Everything is now open. All possibilities are open. We only have really some preliminary evidence in animals, some pilot studies in humans, and I think we're just entering now one of the most exciting phases of biomedical research. Our digestive enzymes cannot be ignored. What we've been thinking about all along, that we only digest food without digesting ourselves, that hypothesis needs to be subject to some very severe testing. And I can't think of any disease where digestive enzymes cannot possibly enter in our bloodstream, including things like aging, and certainly things that you guys are concerned about, anything that has to do with the central nervous system. So, I'm going to close at this particular point, but I want to say one final thing, and that is, in the future, we'll have a new way of greeting each other. Thank you.
We have time for some questions.
I've got a couple of questions, Geert. One is that there seems to be quite a ubiquitous process, which begs the question of, is there actually a normal function for it, right? So generally evolution is lazy and it uses normal processes and sometimes it uses them for other things. So that's the first question. And the other one is do you have a sense of you've talked a lot about your observations in these rats. I thought it was quite interesting that you saw similar magnitude of effects in both your wiki rats and your spontaneously hypertensive rats. And so that suggests that the same thing is happening and that there's maybe not a difference in the two, it's just that the baseline level is different. And so I guess I was just wondering what's the next step back in the process? What is the mechanism by which the this leakage of the enzymes from the digestive system into the blood pressure is happening?
Let me answer in reverse. It turns out the SHR has a genetic defect. And that genetic defect, we know the polymorphisms. It is a chromobranin A. It's a protein that's responsible for packaging various constituents in lysosomal enzymes. That is defective in that. As a result, this animal has, in all major organs that secrete, adrenal gland, pancreas, has a defect in its secretion mechanism. So it just releases material. And so we have first evidence that in the SHR specifically, there's a defect in the adrenal gland, but there's also a defect in the pancreas. And pancreatic digestive enzymes actually leaked in this animal of its lifetime into the portal venous, into the venous system, and it makes its way into the central circulation. And as a result, it leads to an activation of the MMPs, and it's the MMPs that amplify the whole process. In that. So in that particular case, we have a first lead that goes from the genetic defect to an actual physiological manifestations on it. On your first point is the following. Is a release of digestive enzymes. Can I think of any event in life that is vital for us to sit here for survival, where digestive enzymes are needed? Well, the only place I can think of is we all need digestive enzymes even to grow up. We wouldn't be tall persons because we can't absorb nutrients like an amoeba from its surroundings.
through the skin. We have to eat somebody else. The average human being, even if you only need 200 grams per day, which most of us don't, you eat in 1 year your own body weight. You eat another person or another living creature of your own weight. That's a huge amount. This is the largest environmental risk we ever exposed to in our lifetime. Much larger than the microbiome or anything like that. There's no comparison. So we need to digest. We need it for that purpose. Any other purpose I can think of why we need to have digestive enzymes in our bloodstream, I can't think of one. Not one. They're not supposed to be in our tissue. Now, how does evolution work? Well, I'll tell you the obvious. You need to grow up. You need to reproduce. By the time reproduction is done, grandparents, go. We can't have you around anymore because you're using up these nutrients. They're supposed for the next generation. Get away. And nature takes its revenge by saying, yeah, sure, I allowed you to grow up the way you are, but I'm not going to provide you with 100% protection.
Apoptosis is another.
Whole body apoptosis. So what breaks down? What is it about?
It's a mechanics problem.
Yeah.
The only thing that breaks down is that barrier. Once you know, I need to focus on the digestive enzymes, now your focus is super sharp. I need to know where are they, how are they produced, when do I need them, and under what circumstance do I get out. So studying this barrier I showed you is actually at the heart of the problem. What leads to a situation? Eating is a double-sided sword. If I eat too little, no good. If I eat too much, it's no good. So it is a process of optimization now. And in an optimization process, you have to know what's in favor and what's against. That's the fundamental problems of systems engineering. What we haven't paid attention to at all, what is the downside? Every time I eat something, what's the downside? Well, the downside is I damage my intestine. So I have to get nutrients, but I need to minimize the damage to my intestine. So things like, for example, the C-reactive, CR, the calorie restriction, which is a well-known phenomenon in the general field of aging. It's heavily funded by the National Institute of Aging, where you're being told reduce the amount of food you consume. And it's been now shown even in rhesus monkey is actually leading to a prolongation of life and dramatic reduction of all kinds of diseases, including cancer. That's along those lines we need to think. But to optimize this, in my mind, takes an engineering thinking. You have to really think. You have to know what's your upside, what's your downside. You have to know it in quantitative terms, and then you can start to try to optimize what may be the best. It will require a very deep involvement of engineering in this particular field. John?
So, a few years ago, we studied a group of patients who had isolated superior mesenteric ischemia. So, they couldn't dilate after eating. The average person will increase blood flow in the intestines by 4 times after eating protein. These people don't. So, my question would be, have anybody looked at these people? It seems like they should just go downhill like a roller coaster if this is true. Or that also,
right.
if you banned, say, the celiac or superior mesenteric, they should really go down fast. It wouldn't, right? Would that be if this was?
I think it's all a matter of how much. In hemorrhagic shock, we make the whole intestine ischemic.
Oh.
Okay, that's right. If I were to just take a part of the intestine and make it with celiac or with a superior mesenteric artery, it's a question how much. Now keep in mind, I alluded to this briefly, every fluid that comes out of your intestine goes first through the liver. Okay? And the liver absorbs a lot of these, both the enzymes as well as the products they produce, like free fatty acids. But that's a question of quantitative. How much? Okay? In hemorrhagic shock, you can actually show that for about, if I remember the numbers right, about 45 minutes or so, you actually see that these products are being absorbed in the liver. but then suddenly it fails and they appear in the central bloodstream. So the liver is a major protection mechanism that we have, no question about it. And also you have in your bloodstream, I didn't have a lot of time to talk about it, I mentioned briefly the TIMP1. We have anti-proteases in our bloodstream, synthesized in the liver, alpha antitrypsin, for example. All of us have a certain level, and therefore the issue is, okay, if I have suddenly a spurt release from the intestine, do I exceed that buffering capability or I stay below it? If you're below it, chances are that this process is much less consequential. I was talking about the cases where it exceeds it. And then you can indeed end up in a situation where you have the uncontrolled enzymatic activity, and that has all these consequences. So none of these patients have ever been even thought about, let alone studied.
So I really enjoyed that, but I was
Thank
a
you.
little unclear about some of the terminology. So
Yeah.
you talk about shock, hemorrhagic shock, mesenteric ischemia.
Yeah.
I got the impression you were interchanging those terms, whereas I think for clinicians they're all quite different. So,
Yeah, they are.
So that's my first question: is
Right.
all of your experiments were really mesenteric ischemia?
Let me explain. We've studied many different forms of shock. I showed you a few examples. Hemorrhagic shock is the one where you, as I said, the whole intestine, in fact, every organ is ischemic in that particular case. So that serves that purpose.
Well, I think for
If hemorrhagic shock in humans occurs, for example, when a kid in San Diego goes out swimming, gets bitten by a shark, isn't able to come on board very quickly, then you would have a hemorrhagic shock circumstance. So it's not all that prevalent. The splanchnic arterial occlusion is one where we have a local ischemia that we deliberately introduce. That would simulate, for example, reconstructive surgery of the main thoracic aorta in humans. They have to expose an intestine to a period of 15, they tell me even 45 minutes of ischemia, and one is concerned about that. We also studied, for example, in the science paper I cited briefly, we studied various forms of septic shock. And remember, these are all models of septic shock. These are not human septic shocks. And the process that I mentioned, for example, in septic shock with peritonitis or with endotoxin, they all go through the same passage as I showed you here. The major thing that takes place is a breakdown of the mucosal barrier. For example, in endotoxic shock, if you're familiar with this, which is often used as a septic shock model, if you block the digestive enzymes in an animal by just going through this procedure, and you then give a lethal dose of the endotoxin, the animal has a mild inflammatory response and nothing else. Walks away completely from that process, telling us that actually what the endotoxin is doing is it opens up the mucosal barrier. And it's the rushing in of the digestive enzymes that actually kills the animal. So it's the digestive enzymes that kill the animal, not the endotoxin. It's been published and you can read up the details on that. Yeah.
Geert, I have a colleague at Louisville who thinks that Alzheimer's disease is caused by imbalances of the gut biota. So what sorts of interactions between the proteases and the gut biota?
Right. The microbiome, gut microbiome is clearly something to look out for. In the actual killing mechanism, I don't think it participates in a significant way unless somebody has a total overgrowth. But in the control of the permeability, that is a really interesting question because many bacteria are produce amylases, which break down the sugars, and that means they can attack the mucosal barrier, the mucin, on that. And so now it's the question, okay, if you have a local spot somewhere in your intestine and you have a colony there that is producing amylase, are they possibly draining their way through that mucin layer so that you're now exposed to the rest of your digestive enzymes, to your proteases and your lipases? That needs to be studied. I don't know a single study. It's a wonderful idea. But I joined forces with a group on our campus, Rob Knight, perhaps some of you know him, and we decided we were going to start a center for what we call gastrobiome. Okay, it's just a place with words. But the idea is we want to look at the gut as a whole. We're not look at parts of the gut, but let's now give up on this idea and we take the whole gut as it is. So that means everything that's in the gut, food, digestive enzymes, obviously microbiome, and the barrier. They all have to come together. And we need to set up centers across the country. I'm trying to convince the NIH that they should put money into that. Haven't been very successful yet.
Well, one more fraction.
Just curious, probiotics is supposed to be good for you. I'm sure you've thought about it more than me. What's your thoughts on probiotics?
Let me start out with a clear-cut case. In the case of shock, University of Michigan carried out for lots of money a probiotic trial in septic patients. Okay, the same thing which we are trying now by blocking digestive enzymes. That trial joined about another 130 plus trials in the history of medicine, failed completely. Okay, all interventions against shock have failed so far. We don't have a treatment today. So the probiotic context in acute case when you potentially have this massive release of digestive enzymes is way too late. Okay. In chronic situations, it's a different story. If indeed there is a colony, is a certain genome that is producing, say, unusual amounts of amylase, that's sort of a case I can think of. Yes, it could have a chronic effect on your digestive system and on the barrier function of your intestine. But I couldn't point to a particular case at the moment. I have to wait and see. Remember, the probiotic treatment is fashionable. The industry has a conflict of interest in there. They try to sell the thing. But the medical evidence that in double-blind clinical trials that it is effective is few and in between at this particular point. And I would say, if I look at the end, think of this 1 pound of food that you eat every day. Okay? And it comes out the other day. I won't describe in any details on how it's going to come out. But you will agree with me, it's been this wonderful plate you paid $100 for. It comes out in a very different form out of you, okay? Who was responsible for that? Your bacteria or your digestive enzymes? I don't know a single bacteria that would ever be able to take a pound of food and degrade it to such a degree in a period of less than 24 hours. It is your digestive enzymes. They are a player of their own. They have the ability. In fact, we have experiments where we deliberately broke down the barrier in experimental animals, and then we reconstituted the contents of the intestine with and without digestive enzymes. By just returning the normal mixture of digestive enzymes into the lumen of the intestine after you broke down the barrier without doing anything else to the animal, no shock, no other treatment, this animal does not see the sunset. It's dead. So your digestive enzymes have the ability to digest yourself in a period of just a couple of hours. There's one case report in the literature where somebody had the idea and says, I don't have enough digestive enzymes. There's in fact even an industry that tries to sell digestive enzymes, but only as a pill. This guy said, no, pill is not good enough. I need to infuse it directly into my bloodstream. And we referenced somewhere, there was a case report written, but it wasn't written by him in the 40s. So, and indeed, if you take an animal and you infuse digestive enzymes intravenously, depending on how much you infuse, we've done those experiments in the early stage of this research. These animals are dead within just seconds. It's like bitten by a rattlesnake. A rattlesnake also uses, and many other snakes, uses in fact, in part, they use all kinds of other methods, but they use a digestive enzymes that is injected into you from the very beginning, because that's the beginning of the digestion process. So what we have in the pancreas is sitting just in the gland up in these species. And it serves the same purpose. Interesting.
Well, I think we really do need to move on.
Yeah.
So Stephen, if